I've put a ruler here to give you an idea of scale. The two red marks are an inch apart, so this is an inch in diameter. In this particular case, I'm going to be using copper in excess. If we were using nitric acid as our oxidant, we would use nitric acid in very high concentration and in very high excess, and at the end of the reaction, the first step, the solution would be very, very strongly acidic. One of the issues with that is that this reaction goes back and forth between very acidic and very basic conditions, and so in general it's better if, as you move into the next reaction, you get rid of as much of the acid that you're using, for example, or the base that you're using as possible, because you don't want to end up expending a lot of the reagent that you use in the next step, simply neutralizing whatever it is that you carried through as an excess reagent in the previous one. So in this particular case, instead of using nitric acid and carrying through a vast excess of acid, I'm going to use hydrochloric acid, and the hydrochloric acid is going to end up being the limiting reagent. So we should consume most, if not all of it, in the process of the oxidation. And then the solution that we end up with isn't going to be very acidic at all. We might actually, to be honest, have to add a touch of acid just to make sure everything stays in solution before we move on to the next step. If we do that, we need to be able to quantify how much copper we use in a different way, and so I'm going to do that by subtraction. So what I have is a really long piece of copper wire. There's a little spool at the bottom to expose some surface area, and then I'm going to add the chemical reagents to it. I'm going to weigh the entire wire before the reaction, and then I'm going to weigh it when it's done, you know, with its part. And by subtracting those two, I'll know how much copper I ended up putting in the solution. This particular test tube is going to have a little stir bar at the bottom. That's a piece of metal that's wrapped in Teflon, which is an inert plastic. And the idea is that this stirring hot plate has a larger magnet underneath the ceramic part of it. When that magnet underneath there is spun, what happens is that this little magnet inside of the test tube will try to turn in order to stay in alignment. And that allows us to stir a reaction without needing to put like a glass stir rod in it or attend it in any way. You can turn the stirring on and then walk away from it, which is very convenient. So what I'm going to do is weigh the copper wire, and then I'm going to put the copper wire back into the test tube, and then I'm going to add the first of the chemical reagents to it. I'm now going to add 1 milliliter of 6 molar hydrochloric acid. So one of the things that I can observe here is that even though my acid is in contact with the copper and I have a stir bar in there to make sure that the solution is constantly being sort of churned against the surface of the copper, I don't see much of a reaction occurring. If I think about other metals that we've observed, like magnesium or zinc or iron or aluminum, all of those metals react with hydrochloric acid or any acid to produce hydrogen gas, and that puts the metal into solution. But in this particular case, it seems like the hydrochloric acid isn't sufficiently powerful as an oxidizing agent to get the copper to go into solution. So this is one of the reasons that traditionally you might use nitric acid instead, right? Is that nitric acid is both a very strong acid and also a very powerful oxidizing agent. And so if you use nitric acid, part of the nitric acid will act as the oxidizer that will take the electrons from the copper, and the rest of it will act as a counter ion when the copper goes into solution. In this particular case, the hydrochloric acid itself is just not sufficiently powerful. What I'm going to do to get the copper to go into solution is I'm going to add a more powerful oxidizing agent. In this case, I'm going to use hydrogen peroxide. And the reason is because under acidic conditions, hydrogen peroxide is a really powerful oxidizing agent. And the waste products that it produces if it overreacts or if it decomposes without oxidizing something are oxygen, gas, and water. One of the challenges that exists working with nitric acid is that in general, when you do nitric acid oxidations, you end up producing nitrogen oxide or nitrogen dioxide, and those gases are toxic, and so they're noxious. You have to do this kind of reaction in a fume hood, and it's just inconvenient. So I'm just going to add 1 drop of 30% peroxide to begin with. Now, as you can see, the moment that I added 1 drop of 30% peroxide, there's a color change, and that's indicating to us that this is a successful oxidation. Something is going into solution that is colored, and that's probably not something which is just oxygen atoms or hydrogen atoms or chlorine atoms even. The only species that could really be part of this reaction system that would make a colored solution would be copper ions. We know that copper ions in a lot of cases are either green or blue, and so the addition of just a drop of peroxide leading to this green solution indicates that we're doing a decent job of oxidizing that copper. So I'm going to add a few more drops to that. Now, this peroxide that I'm adding is a 30% solution, which is about 10 times as concentrated as the standard solutions you might get in a convenience store. It is itself coming directly from the refrigerator because we want to make sure that it stays stable as long as it can be for us to use it. So some of the fog that you might see in there could be a result of temperature differences between the different reagents as they're mixing together. It is kind of important to note that in this particular case, the reaction solution that we see is kind of a deep green color as opposed to a blue color that you might think of as traditionally coming from a copper complex. So as a color contrast, what I've put here is a 3 molar copper 2 chloride solution that I prepared independently, and over here is a 1 molar copper 2 sulfate solution. So one of the things that you can see about these is that copper solutions can have different colors. Traditionally, we think of them as being blue, so a copper 2 sulfate or a copper 2 nitrate solution will have this very distinctive blue color. But if we have really, really concentrated solutions of copper 2 chloride, they have a tendency to look a dark green like this, and that's not a result of this reaction failing or not working properly. It's actually a characteristic of this particular solution at that concentration. In this case, one of the things that we can think about is that if this species has a different intrinsic characteristic, if there's something in there that's absorbing light and it's a different color, then that must mean that there's a different species present. So one of the things that we need to think about is that we have copper 2 ions in a copper 2 sulfate or a copper 2 nitrate solution that traditionally have this very distinct blue color. And then in this case, if we have copper 2 ions that are in very concentrated solutions with chloride, we end up having something which looks green. And if you look here in this case, it actually changes to almost a brown color, indicating that there is definitely some other version of the copper ion that's in solution. And exactly what the nature of that is, you wouldn't be able to tell simply from the difference in color. But since it involves having a lot of chloride ions as opposed to a lot of sulfates or a lot of nitrates, it's not sort of inconsistent with that to say that it's possible when you have solutions that have a lot of chloride in them. So in this case, this is a 3 molar copper 2 chloride solution, which is approximately the same concentration that this would form. If you have a lot of copper and a lot of chloride ions and they're all stuffed together in the same place, that you might end up with ions that have chloride stuck to the copper and floating around independently. So those things would be like CuCl4 2-. These things are called complex ions, and they're just, it's another form of copper that's floating around in solution. But one of the things we do want to make sure is that before we move on to the next step of the reaction, is that we want to make sure that all of the copper ions that we have in solution, or as close to all of them as we can, are of the same type. And so in order to do that, what I'm going to do is take the copper out of the solution, and then I'm going to add just a little bit more acid to it and a little bit more peroxide in order to make sure that everything has been fully oxidized and fully oxidized to the same form. So the first thing I'm going to do is just run a couple of drops of acid down the copper that's left to dissolve off anything that might be in there. And so now you can see that the copper that's left over has a pretty bright color. Now, interestingly enough, my solution still has this very dark brown. And one of the reasons can be that if you have enough chloride ions in the solution, if it's concentrated enough, then not only can the acid act as an oxidizing agent, not only can the peroxide act as an oxidizing agent, but it's actually possible for copper 2 ions that are in the solution to act as an oxidizing agent, and they can oxidize the copper metal. So you get one copper 2 ion reacting with one atom of copper metal to form 2 copper 1 ions. Normally, copper 1 ions wouldn't be stable in solution, but in this particular case, since it was 6 molar in chloride ion, we can get these complex ions of copper 1 also, where you have copper 1 with 2 or 3 or 4 chloride ions stuck to it, and those complex ions are also soluble. So we could potentially have a solution here that has some copper 2 ions and has some copper 1 ions. And as we move on to the next step, we want to make sure that everything is in the form of a copper 2 ion so that we can do the next reaction. So what I'm going to do is just add another drop of peroxide. And what you can see is that that brown color seems to go away, indicating that some chemical reaction has happened with that peroxide. We haven't seen a whole lot of bubbles forming right? They do form a little bit, but there's definitely been a chemical reaction between the hydrogen peroxide and something which was in the solution. And what you can see is that our color is now changing, so it's now much closer to the color that we expect for a 3 molar copper 2 chloride solution. So again, we started with a solution that had some copper oxidized into it using the hydrochloric acid and the peroxide. It was probably a mixture of copper 2 and copper 1. And then by adding that last little bit of peroxide under acidic conditions, we were able to get this solution to be mostly, if not all, in the copper 2 condition. Now, if I add some water to it, as we dilute it out, what you can see is that it's not just that the intensity of the color changes, but the color itself is changing, right? And that's an indication to us that the chloride doesn't really want to stick to the copper very strongly, which makes sense, right? Because copper 2 chloride is soluble in solution. So we don't expect the equilibrium that sticks chloride on copper to form those complex ions to be very favorable. It should only exist, right? That species CuCl4 2- probably should only exist and only be favorable under circumstances where the chloride ion is very, very high in concentration. So if we go from the solution as we started off, where it was just a couple of milliliters at the bottom, and that was maybe 3 molar, and we've now increased the volume maybe 10 times that much, we've taken the chloride ion concentration probably below 1 molar. That means that the species that tend to make this green color are going to disappear. They're not favored by the equilibrium. And so then we see what we expect copper solutions to look like.